Hello, class. This is Mr. Mormon. This is your test review for the unit test on cells and the microscope. Let's start with the microscope. We're going to talk about safety and the proper use of the microscope. Remember, always carry a microscope with two hands, one on the arm and one under the base. Set it flat on the table with the arm facing you. Look from the side when changing lenses and always focus by moving the lenses away from the slide so that you do not hit. This can scratch the lens and break the slide. This is more of a problem when you're using the high-powered lens because it's much longer. Remember to always use the coarse adjustment knob when you're focusing under low power. After you focus under low power first, then you're going to switch to medium or high power and use the fine adjustment knob. A microscope does three things to an image. It increases magnification, making things look bigger. It increases resolution, making them sharper, so you can see more detail. And finally, it reverses the image, making it upside down and backwards. We learned in class that we can use the microscope to estimate the size of microscopic organisms. To do this, we must measure the field of view. We're going to use either a thin, clear ruler or a grid slide. This is going to give us measurement in millimeters. Very often we want to measure or estimate in microns or micrometers. Remember that 1 millimeter is equal to 1,000 microns or micrometers. Okay, in this unit we also learned how to make wet mount slides. You place a drop of water on the slide, then your specimen, and a cover slip. Put the cover slip down at a 45-degree angle, let it down gently to eliminate air bubbles as much as possible. If you're going to use stain, which we sometimes add to make certain cells or cell parts stand out better, you put the drop of stain at the edge of the cover slip and then put paper towel on the other side of the cover slip and it will draw the stain up underneath. We learned all the different organelles that you'd find in a typical cell. Remember that like the organs in a multicellular organism, organelles perform all of the life functions or metabolic processes that keep a cell alive. Remember that metabolism is the sum of all the chemical activities keeping an organism alive. We also learned about the levels of organization in a living thing. From smallest to largest, cells are grouped together. The same kind of cells make a tissue. Several different tissues working together to perform a job would be an organ. Organs make up organ systems. Put all the organ systems together and you have an organism. A tissue is a group of similar cells all working together to do a particular job. You can talk about muscle as being a tissue. Bone is a tissue. Brain is a tissue. Even blood is considered a tissue because it's made up of different types of cells, like red blood cells, white blood cells, and platelets, all working together to do different jobs. You should be able to fill in this table from what we've learned. Why don't you pause for a minute and see if you can fill this out. It's also on the review sheet. And here are your answers. I would also expect you to be able to label diagrams of typical cells, animal cell and plant cells. And there are the answers. You should be able to describe the differences between plant and animal cells. Plant cells have a cell wall, a thick, rigid cell wall around the outside made out of cellulose. Animal cells don't. Plant cells have chloroplasts for photosynthesis. Animal cells don't. Plant cells have a large water vacuole. Animal cells have vacuoles, but not a large water vacuole. You should know about ribosomes. They're very small organelles. They float in the cytoplasm or are attached to the endoplasmic reticulum. We would then call that rough endoplasmic reticulum. And their job is to make proteins from the instructions on the DNA in the nucleus. You should also know about the mitochondria. These organelles are the powerhouse of the cell. Remember, we called them the mighty mitochondria. They take in glucose and oxygen and release energy for the cell to use. This is called cellular respiration. They are rod or bean shaped with a folded membrane inside to help you identify them. You should understand the basics of protein synthesis. You should know that DNA in the nucleus is a blueprint for making proteins. Amino acids are put together in the right order to build a protein in a special shape in order to do its special job. The proteins are assembled by ribosomes in the cytoplasm or attached to the endoplasmic reticulum's wall. Information is copied from DNA and transferred to the ribosomes by RNA. Some proteins are structural, like muscle, bone, and hair. Other proteins control all of the chemical reactions keeping an organism alive. These would be called enzymes. Again, you should know that DNA is located in the nucleus of a cell. Here's a model of what DNA might look like. And if we were to talk about some of the vocabulary that goes along with DNA, nitrogenous bases are the letters in the DNA alphabet: A, T, C, and G. Adenine, thymine, cytosine, guanine. Those are the colored little bars you see in the rungs of the DNA ladder. Now, nucleotide would include those nitrogenous bases plus the pieces that hold it together in this kind of twisty spine here. There's 5-carbon sugars and phosphates that hold it together as well. A short DNA strand would be the next largest thing. Now, a gene is going to be a pretty big piece of DNA. It's got the instructions to build a complete protein. It would probably be hundreds or thousands of bases long. And a chromosome is going to be even bigger than that. Chromosomes can contain thousands of genes. If you think about a human, there's over 100,000 genes in a human cell, but only 46 chromosomes. So each chromosome is going to have many, many, many genes. Again, a segment of DNA with information to make a complete protein is called a gene. Now, proteins are made of the building blocks called amino acids. When we eat protein, they get digested into amino acids and then travel through the blood to cells that pick them up and use them to synthesize new proteins. So remember, taking large molecules and breaking them into small ones is digestion. Synthesis is the opposite of that, taking small molecules, putting them together to make larger molecules. While we're talking about building blocks, remember that the building blocks of starches are sugars, and the building blocks of nucleic acids, like DNA and RNA, are called nucleotides. We've learned a whole lot about photosynthesis and respiration. Let's talk about autotrophic nutrition first. Autotrophic nutrition, this is where organisms make their own food. Now, usually we're talking about photosynthesis when we're talking about autotrophic nutrition. This is making food using sunlight energy. Plants do this and provide energy to almost every ecosystem. We also talked a little bit about chemosynthesis. This is what happens at those deep sea hydrothermal vents at the bottom of the ocean, where those bacteria are using chemicals and the heat bubbling out from those hydrothermal vents to make the carbohydrates or the sugars that become the base of those food chains. But generally, we're talking about photosynthesis when we're talking about autotrophic nutrition. Now, remember that the purpose of photosynthesis is not to make oxygen. Students always make that mistake. The purpose is to capture the energy in sunlight and store it in the chemical bonds of glucose. The oxygen is just a byproduct. The plants don't even want it, and they dump it into the atmosphere, which is good for all us oxygen breathers, but it's definitely not the reason the plants are doing photosynthesis. Now let's talk about heterotrophic nutrition. Heterotrophs cannot make their own food, so they need to eat. They're going to be eating plants or other animals to get the energy they need. The purpose of respiration is to release that energy that was stored in the chemical bonds of glucose during photosynthesis. It's the exact opposite of the reaction to photosynthesis. The energy released from glucose is stored in the ATP molecule and used to power the cell's day-to-day activities. So the energy that sustains almost all life on this planet comes from sunlight energy captured during photosynthesis. Here we can see an overview of the two reactions. Raw materials will be over here to the left of the arrows. For photosynthesis, it's carbon dioxide and water. For cellular respiration, it's this glucose molecule and oxygen. Products are over here on the right. For photosynthesis, we're producing that glucose molecule. Remember, the whole idea is getting that sunlight energy trapped in those bonds. The products of respiration are carbon dioxide and water. What we've done is broken up the glucose molecule and let that energy out. It then gets stored as ATP. Remember that photosynthesis happens in chloroplasts in plant cells, usually in the leaves, and it only works when the sun is out. Cellular respiration happens in the mighty mitochondria, and it's going to happen day and night in all living cells, both plant and animal. In this unit, we also learned about fermentation. We saw a demonstration of fermentation. We could also call it anaerobic respiration. This also releases energy stored in the chemical bonds of sugar, but it does not require oxygen. Yeast and bacteria do this all of the time. Respiration is too difficult for most of them, so they do alcoholic fermentation. Now, your muscle cells will sometimes switch from respiration to lactic acid fermentation if they are not getting enough oxygen during times of extreme exercise. That's the burning feeling you feel when doing certain extreme exercises. It's the buildup of lactic acid from the lactic acid fermentation. When you rest, that lactic acid gets cleared away when you recover. Okay, in this unit we learned a lot about the cell membrane, particularly about transport and how cells communicate. Let's start with transport. You need to understand the different types of transport. There's passive transport and active transport, and let's compare them here. Passive transport would be things like diffusion and osmosis. Osmosis is just the diffusion of water across a membrane, but it's a kind of diffusion. Active transport is just active transport. In passive transport, molecules are moving down the concentration gradient. In active transport, they're moving up. In other words, in passive transport, we're going from high to low concentration, and in active transport, we're going from low to high. Passive transport, like diffusion and osmosis, require no energy, whereas active transport does require energy. The analogy we used was like sledding down a hill. That requires no energy, but of course it takes energy to go back up the hill. Remember that a molecule's size determines if it will diffuse across a membrane. Some molecules may be too large to diffuse across a cell's membrane. The diffusion across a membrane lab showed us that water and glucose were small enough to diffuse. Starch was too big. We learned that embedded in the cell membrane are these special proteins that are helping with transport. This would be a channel protein or a protein channel, which is basically just an opening that's going to help larger molecules pass through. This is facilitated diffusion. To facilitate means make something easier. Facilitated diffusion, it's still diffusion. This does not require energy. These molecules are going from high concentration to low concentration, does not require any energy. This is a special protein molecule. It's a transport protein, and it requires ATP to force these molecules to go against the concentration gradient. That would be active transport. Remember, in that same lab, we also looked at what happens to cells when you put them in salt water. Salt water is hypertonic, meaning there's a lot of solute, a lot of stuff dissolved in it. So it's not 100% water. As a matter of fact, the percentage of water is less than what's inside the cell. So this forces water to diffuse out of the cells. Here in 100% distilled water, the cells are going to be totally full of water. The water would be moving into these cells by diffusion because it would be high on the outside, 100% on the outside, less on the inside, so the water will diffuse in and it will swell up, filling with water. Now, an animal cell, which does not have this rigid cell wall around the outside, may actually swell and burst. If you put red blood cells in distilled water, they swell up so much that they might even burst. Okay, let's talk about how cells communicate. Cells communicate with chemical messages. These simplified diagrams show the different ways that cells can communicate. The important thing to remember in all these cases, it depends on the cell receiving the information, having this receptor molecule to receive the information. In all of these, we see this little green shaped thing that is receiving the message. Here we see in the endocrine system hormones. These are chemical messages that are sent from glands into the blood. They travel through the body, and they're received by cells that have the proper receptor molecules. They will be then a target cell and will receive that message. Neurons in your brain and nerves communicate the same way. The gap in between the two is called a synapse. And the message is electrical when it travels through the neuron, but when it gets to the synapse, it's got to jump across, and it's a chemical message that jumps across. Those chemicals would be called neurotransmitters. This is the area where drugs are going to work. Like a pain-killing drug would either gobble up the neurotransmitters or block up the receptor molecule, so it would stop the pain message. And some cells that are right next to each other can communicate just with the chemicals on their surface. Again, cells communicate chemically. Okay, let's talk a little bit about mitosis. Mitosis is cell division. Unicellular organisms like bacteria do it to reproduce. Multicellular organisms use mitosis for growth and repair. When cells wear out and die, or if there's damage like a cut or scrape or broken bone, we're going to get new cells by mitosis. In mitosis, the first thing that happens is the DNA gets replicated or copied. Each daughter cell gets an identical set of the DNA, which is also identical to the original cell. We end up here with clone. Multicellular organisms do it for growth and repair. Cancer is mitosis gone mad. It's uncontrolled growth of abnormal cells. It starts with a mutation in DNA. So here we have normal cells. Something happens to one of these cells. Could be from radiation, could be from chemicals and cigarette smoke, whatever. Anything that can mess up DNA can lead to cancer. It could be a virus even. The DNA in that cell gets messed up and it starts dividing out of control. Here we have a mass of these abnormal cells. That's a tumor. Sometimes even pieces of the tumor can break off, and that's one of the ways that cancer can spread. That cancer cell would break off, go in the blood, and travel to another organ. Okay, you should know the difference between sexual reproduction and asexual reproduction. So let's compare and contrast. They're the same because they're both creating new members of the species. That's reproduction. Sexual reproduction involves two parents, where asexual reproduction only involves one. As we said before, asexual reproduction leads to clones. The offspring and parents are identical, and all the offspring are the same. You have variation in sexual reproduction. Sexual reproduction involves the female sex cell, which is the egg, combining with the male sex cell, which is the sperm. It's called fertilization when the sperm and egg come together, joining the DNA from mother and father to create the offspring. There is nothing like that in asexual reproduction. Okay, we could talk about several different types of asexual reproduction. There's just regular mitosis. This is sometimes called binary fusion. I'm sorry, binary fission. This is where a cell goes through mitosis and splits to become two cells. This is what bacteria and lots of microscopic organisms do. Then there's budding, like we see happening here with these yeast cells, or this hydra, which is a water organism. The parent plant just grows the parent, it's actually an animal, grows a new offspring right from it. There's just one parent. These yeast cells are creating new little baby yeast cells. We can see asexual reproduction happens in many different kinds of plants. Also, here's a strawberry plant which sends out a runner and a new plant grows from it. It's going to be genetically exactly the same as the parent plant. An onion bulb or tubers can be taken from a plant and replanted, and it will grow a whole new plant that's going to be genetically identical to the plant you started with. Okay, another concept you should remember is that an experimental control is one part of the experiment where you leave the manipulated or independent variable out to see what would happen. This is your normal setup. We do this so that we have something to compare. And let's wrap this video up and we'll talk a little bit about homeostasis and regulation. Homeostasis means maintaining stable internal conditions. Everything needs to be kept at the proper level. Your brain and body are constantly monitoring your body temperature, your blood glucose, your blood oxygen levels, and controlling them, keeping things at their proper level. Regulation means living things respond to changes in their environment. Again, this is related to homeostasis, keeping everything stable and at its proper level. There are many things your body will do to regulate or change things as the environment changes. If your blood sugar level is too high, you will produce more insulin. This will bring blood sugar levels down. If carbon dioxide levels in your blood are too high, your breathing rate will increase. You will get rid of that extra carbon dioxide and get more oxygen. When your heart rate increases, like when you're exercising, the level of oxygen to your cell will go up. Another example of regulation we can find in a leaf. These guard cells, these are openings at the bottom of the leaf, allow carbon dioxide into the leaf cell and oxygen out. It's also controlling the amount of water loss from the plants. Here we see some actual pictures of the guard cells and the stomata, and here's a science, an artist drawing. Again, these stomata can open and close. The guard cells can bend and change their shape, controlling the movement of carbon dioxide in, oxygen out, and limiting the amount of water the cell loses. This is regulating the water loss from the plants. Okay, I think that's definitely enough for this video. If you have any questions, please ask me tomorrow before the test.